Hello to all the HubLE audience. My name is Fatemeh Malekipour. I'm a postdoctoral research fellow at the University of Melbourne, Australia. I'm also a member of Meet the First Author committee at HubLE. I'm here today for an edition of Meet the First Author with Raneem Haddara. We will be talking about their paper titled Anterior Cruciate Ligament Agonist and Antagonist Muscle Force Differences Between Males and Females During Perturbed Walking, which was published in Journal of Biomechanics. So Raneem, welcome to HubLE. And to start with, could you tell us a summary of your work and how it fits into the big picture of this field, which I guess is ACL injury, right?
Yeah, so this paper was the first paper I published from my PhD. Basically what it was is I was really interested in trying to figure out, you know, the ACL is such an enigma until this day, and a lot of people are having trouble trying to figure out, you know, how to stop it, why it happens, and whatnot. And one thing that really was really interesting to me is that females are a lot more likely to have their ACL torn. Yeah. And I was wondering, why is that? I mean, why are females about, you know, up to nine times more likely? So I started looking at papers. There were lots of papers that were looking at differences between males and females. Lots of people didn't even find any differences, and some people just found small differences. And one of the main things that I found is a lot of these studies didn't really try to replicate a real life environment. Most of these motions were planned movements. So telling the subject, you know, jump or land, and then look at the biomechanics of that. But in a real life scenario, a person doesn't really have time to plan for these movements, right?
Yeah.
So I started looking at papers that compared anticipated versus unanticipated movements, and they actually found differences within the same people when they can planned a movement versus not plan a movement.
Right.
And their biomechanics were more injurious when they did the unplanned maneuvers. And the unplanned maneuvers would simply be, you know, they know they're about to jump, they just wait for a signal and then they jump.
Yeah.
But basically it tries to remove that planning time for a person to do something. So I thought, okay, how can I maybe take that a step further? Which is where the perturbations come in. So I figured, you know, perturbations are completely unanticipated. The person is just walking and then they're suddenly perturbed. And I found a paper that said about 96 or 97% of people that tore their ACL were perturbed one way or another.
Yeah.
So that's kind of what made me start to look at it. So I started looking at just simple walking and perturb them and see what their muscle patterns were like, the muscle forces, like how do they recover from that? And it was very interesting to me that females and males recover very differently. And what females do is they recruit muscles that are more likely to cause the tibia from translating forward. So you have your femur and your tibia, and it causes, so when your quadriceps, for example, contract at a very high force, it can cause your tibia to translate forward. It's called anterior tibial translation. And that could tear the ACL. And females had much higher forces than males in that sense. They also had higher gastrocnemius, which is also known as an ACL antagonist. And they had lower soleus muscle forces, which is supposed to protect the ACL. So it seems like females were recruiting the wrong muscles at the wrong time, which is why they're likely to injure their ACL.
Oh, that's great finding.
Yeah, it was really interesting. I didn't expect that, but yeah.
Oh, wow, that's amazing. So as we talked before, you have an amazing setup to perform this study, perturbations and everything. Did you want to share that with us?
Yeah, so let me share my screen. I was very lucky to have access to this lab. So this is my lab right here. So it's called the Karen lab, and Karen is just short for computer-assisted rehab environment. And it is the first of its kind in Australia. So we were very lucky to have it at Melbourne. And basically what it is, it's a pretty much a motion capture lab, but it's a fancy version of it. So you have the semi-immersive screen right in front of you. The person is harnessed, And then the platform that they're walking on moves in all 6 degrees of freedom. So you can pretty much employ different kinds of perturbations. And then the cool thing as well is
environment, yeah.
Yes, in a safe environment,
Exactly.
Obviously. So, yeah. And so you can see the split belt treadmill here. So the cool thing about that is when the person is walking, you can actually control either belt separately. So you can make them go at different speeds, which is kind of what I did as well. So you can make one belt go faster than the other or slower than the other. And it kind of mimics like a slip or a trip, which could happen in a sporting field. So yeah, that's pretty much what I did.
Awesome.
So I took this data, so the kinematics and the force plate data from the, there are force plates in the treadmill. So I took those and then I used OpenSim for musculoskeletal modeling. And for those of you that are not really familiar with musculoskeletal modeling, here's a very simple version of it that is actually built into the Karen. So you can see over here, the subject has these markers on his body, and then this avatar, for lack of a better word, in front of him is kind of mimicking those same exact movements. And this specific model calculates muscle forces in real time. So it's a very simple calculation, but you could see the muscles turning green. And that's kind of, you know, the muscles that are producing the force at that point in time.
Beautiful,
Yeah. So the perturbation that I had in the paper was a deceleration perturbation. So basically when the person was walking, I made one treadmill, one belt suddenly come to a stop and decelerate. And this is a video of a person being decelerated. So you can see once at heel strike, they lose balance and then they regain balance. So that's kind of where I was looking at the muscle forces, what they were doing at that point in time.
Great. That's okay.
And did you want to talk about statistical parametric mapping?
Yeah, I'm a little conscious of the time, but yeah, go ahead.
You want me to? Okay,
this is
so
a nice, this is a nice statistical method, so I think it's worth sharing.
Yeah, I'll show it really fast, but basically, I discovered SPM at ISB, was it last year or the year before? I can't remember.
Okay.
But basically what it is it's a different way of looking at biomechanical data. So instead of taking scalar, like taking out specific numbers, so looking at like maximum joint angle or minimum joint angle, you kind of look at the graph as a whole. So in this case, this is the gait cycle, this is the knee extension flexion angle, and basically it compares the graph as a whole. So it does this cool hypothesis test on the side, and then it compares it. So in this case, from about 0 to 10% of the stance phase, the P was less than 0.01. So you're able to kind of see that, and you're able to catch things that you might not have been able to catch just by looking at specific scalar quantities.
Awesome. Yeah, this is great. Yeah, thanks for
yeah, no worries.
A brief and nice explanation.
I tried to make it really fast.
No, this is great. Thank you very much, Raneem. And as the final question, in terms of your career goal, where do you think this paper fits, and how is this going to help you achieve your aims and career goals?
Well, it is my dream to stay in academia. So obviously this paper is going to bring me this much closer to becoming a professor sometime in the very far away future. But this paper has been a good talking point in my interviews for my postdocs. So that's been great. I mean, I officially haven't started yet, but eventually I'm hoping to start a postdoc in September.
Oh, so wow. Great.
I'm really excited about that. So, yeah. That's awesome. And it was all because of this paper. So it was great. So thanks.
Awesome. That's awesome. Great. Thank you very much. This is a lovely work, and we definitely hope to see a lot more from you. Yeah, and even during your postdoc. Thanks very much. Thank you.
Thanks so much for having me, Fatemeh.
Yeah, sure. Thanks for being with us today. Awesome. Great. Thank you.
Bye.